Welcome to Inflammation Nation. My name is Gráinne O'Leary. Inflammation Nation is a podcast from Arthritis Ireland aimed at increasing awareness and understanding of arthritis and related conditions. Today I am joined by Professor Gerry Wilson and Professor Ursula Fearon, the Arthritis Ireland Chairs of Rheumatology. Professor Gerry Wilson is a practicing consultant rheumatologist with clinics in the Mater and St Vincent's hospitals. He was appointed to the Arthritis Ireland UCD Chair of Rheumatology in 2013 and leads the UCD Centre for Arthritis Research. Professor Ursula Fearon was appointed Arthritis Ireland Chair of Molecular Rheumatology in Trinity College Dublin in December 2016. Hello to you both, you're very welcome.
Thank you.
If I can turn to you maybe, Gerry, first. You were Professor of Rheumatology and Head of the Sheffield ULAR Centre of Excellence for Rheumatology before your move to UCD in 2013. You know, what was it that enticed you to take up the position of the Arthritis Ireland Chair of Rheumatology in UCD?
Well, that's a good question, Gráinne, and I guess there's a mixture of reasons. So the personal reason is it was the right time, and number of reasons for saying that. I was at a stage where a lot of my contemporaries who were appointed at the same time in Sheffield were moving on to more senior positions. The other thing, very personal thing, is my children had finished school, and that's a good time. We travelled a lot before the kids went to secondary school, and we were in the States as well as in Scotland and England. And so we'd moved around quite a bit, and then we moved back to Sheffield, and really we had to stay there while the kids were in secondary school. So once that stopped, that was the other major personal reason. And then the other major reason is that there was a great opportunity with the Arthritis Ireland Chair to undertake high quality research, and particularly, as I've said to lots of people, Ireland is a great place for rheumatology research. It has historically been a great place to do research. It's very productive, great strengths across the country and the universities. And the consultants particularly are very many of them are interested in research and have actually undertaken research during their training, and they're very keen to contribute. And I knew a lot of people in Ireland before I moved across. And so there were lots of, it was just the right time and it was the right opportunity to move at that time in 2013.
And Ursula, if I can turn to you. You know, your field is molecular research. Well, first of all, maybe can you explain to us what does that involve? And also what led you to pursue a career in this field?
So I'm a scientist, so not a clinician at all. So I think I was always loved science, and you know, it was fed by a brilliant biology teacher where I went to school in Muckross, and she kind of just made it very interesting, and she applied all the science to human diseases. So then I went into UCD, did a degree in biochemistry, but I think what really got my interest in translational research was my fourth year project, because in biochemistry the majority of the projects were really hardcore biochemistry, molecular biochemistry, but none of them were applied to human disease. And the only one that had a project was Professor Gethin McBain, who had a project on Alzheimer's disease. So I applied for her, and it was the first time that actually I suddenly realised what we were doing in the lab could actually translate over to a patient. So therefore that kind of instilled in me that I wanted to be in that area of research. And so then I moved down to Vincent's to do my PhD in a completely different area from rheumatology, endocrinology. And my project was on looking at what factors influence infertility and polycystic ovary syndrome. So how I ended up in rheumatology is a bit strange, but I think in science, you have the skills to transfer. You do an awful lot of the same methodology no matter what, I suppose, disease you're in. So I moved to Leeds in 1997. And I was a bit naive because I knew nothing about rheumatology. And Gerry will probably confirm this, that it was probably starting to be the most exciting time in rheumatology research. Because, you know, if you look at papers 30 years ago, you know, and if you talk to a clinician from 30 years ago, they would say half the patients in their waiting room were in wheelchairs because there was no good medication for patients with inflammatory disease or any kind of form of arthritis diseases. So I moved to Leeds, where Paul Emery was setting up the unit. It's probably one of the biggest clinical trial units in the world now. And basically I arrived in when all these new biologics were being tested both in the lab and in the clinic. And so I literally got hooked within 2 months in rheumatology, and I've been there ever since. So that's, what started it all. And I think where I really found it kind of got an insight into the patient aspect of rheumatology was because the scientists were never allowed to meet the patients. We're a bit scary to the patients because, we want, samples, we want blood and we want tissue.
Want something from them.
Yeah. So actually there was one case study in Leeds, which I was involved with, and she was a 25-year-old girl who was in a wheelchair. They tried every, medication and nothing worked. And they did an autologous stem cell transplant, which is, is quite a tricky, as Gerry would say, it can be quite risky. And at baseline, when she was in the wheelchair, I was given a biopsy, and we looked at the biopsy full of, bad immune cells. And then 3 months after the stem cell transplantation, she was walking mountains. And I got another biopsy, and there wasn't an immune cell in sight. By 6 months, she was walking mountains still, but I got a biopsy and I could see cells back in her joint. And by 9 months, she was back in a wheelchair. So that had a real impact on me as to, we really need to understand what's going on with these patients. And that patient said she would have gone back and done the stem cell transplantation again if she could, just to get 6 months relief from the pain. So that really got my passion going in rheumatology, and I've been in it for 20 years now.
And of course, you've both become the Arthritis Ireland chairs of rheumatology in Trinity and UCD, respectively. And that was a long-term goal of Arthritis Ireland in terms of an investment in rheumatology research and investment in the infrastructure in rheumatology research in Ireland. What has that investment in those chairs of rheumatology meant for you in your role and, and for rheumatology research in general in this country?
Well, I think it's important when you start a new position to start with, running as quickly as you can. And the funding that came with the two chairs allowed, I think, both of us to develop research groups very quickly and to, undertake. And we've both been, I think, reasonably productive with regards to findings and, publications and dissemination of our results and finding new things. And so the funding was very attractive because, as Ursula will tell you, it's very tight and very tough to get funding. Most applications, if you're a 15% hit rate, it's about average. So 5 out of 6 times you put a grant application in for funding, it's rejected. And increasingly these days, it gets top marks. And it's put on reserve lists, and it's very tough. And Ireland is a bit now like what it used to be in England, the MRC, which is the toughest and most prestigious funding agency there. Frequently you'd get 90% marks from all the reviewers and not get funded. And that's what's happening here at the moment. So the funding that came with the Arthritis Ireland chairs really helped us to get up and running, going and develop some critical mass and take it forward from there. And without that, it would have been a real struggle to get research going, I think.
Yeah, no, I totally agree with that. And I was in a slightly worse situation than Gerry because Trinity was an empty space. So I walked into a lab in TBSI and I had a beautiful new lab, but there wasn't one piece of equipment, not one bit of consumables. And so if I didn't have the funding support of Arthritis Ireland, there was no point in making me a chair and having the funding, for my position. If I didn't have, support and funding for other people and for the actual equipment that we used to do the research, you wouldn't be able to do the research, you wouldn't be able to do the translation of research, and therefore you wouldn't get the outputs, which could have some impact on patients down the line, and you wouldn't get the publication. So it's kind of a vicious circle that if you don't get the publications, you don't get the actual research grants. So if I'd gone over to Trinity to that empty space and I'd had to, bring in, if I had no support, I would have had probably spent 2 or 3 years just trying to get the lab up and running. So the fact that I had that funding support, it meant that I could get the lab up and running really, really quickly. And therefore we got an awful lot of our initial results within a year or two, and then it feeds into the whole process.
Which enables more things to happen.
Exactly, yeah.
It's a sort of virtuous circle. You have funding, then you get grants, publications, and then you can apply for more funding. And it's that circle, and you have to maintain both components of it in the medium to long term, and that's a tough thing.
It's a bit like your last book. Everyone says you're as good as your last publication. So if you published one 10 years ago,
So I think we've touched on this already, but it is, a really vibrant time for rheumatology and immunology. There's been huge advances in clinical therapeutic strategies. Can you take us through some of those key developments that have taken place in recent years? Just in terms of what research has happened and I suppose the resulting therapies that have come out of that and how that's impacted patients.
Well, certainly and we can divide the different types of arthritis into mainly inflammatory or autoimmune, which are rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis, and then the ones that are not inflammatory, like osteoarthritis. So great advances have been made in inflammatory arthritis for a whole series of reasons, and it's getting tissue and patients contributing to studies, which is key, and getting samples in and doing experiments. And if you look back 30 years, that's what was key to identifying TNF as the target, and the other subsequent biological agents. Those were quite fundamental research that relied on lab-based experiments from tissue from patients with rheumatoid arthritis, but the same with psoriasis. And so there have been great advances based on simple experimental procedures, and that continues. So for the inflammatory types of arthritis, the latest developments in the clinics are the new drugs targeting a new pathway called JAK inhibitors, which we're using for most of the inflammatory types of arthritis. And again, they're they've really, as Ursula was saying, the revolution in outcomes for inflammatory arthritis is amazing. And when I started doing rheumatology, it was sometimes most patients came up in wheelchairs and they had uncontrolled inflammation, and the treatments were as bad as the condition, high dose steroids, gold and histamine, and they had awful side effects, and patients required monthly blood monitoring coming up to clinic, and that's been completely revolutionized. But we clearly we're not we can't switch. We can't predict who will get rheumatoid arthritis at the moment, and everyone is different. And so I don't think we're close to having a cure, because everyone is different. We need to know that personalised thing to try and know the best treatment for someone and how to switch things off. So, but there's great progress for inflammatory arthritis. The big area that of need, particularly is osteoarthritis, which affects. I think I saw figures that about 1 million people are close to that in Ireland. And we really don't have any disease-modifying drugs.
Why do you think that is, given that osteoarthritis is, it's the one that affects, if you look at a population level, it affects far more people in our population than inflammatory arthritis?
It's because it's such a diverse condition. I think it can affect different joints for different reasons. The knee, for example, can be involved because of trauma. Someone's played sport and have had a minor rupture of a meniscus or a tendon in the knee, and that can cause it. Or it can be family history. It can be in other inflammatory conditions. And if you look just at the genetics, the genetics of hand osteoarthritis, which is common, is completely different from the genetics of developing osteoarthritis in the knee or the hip. And so it's very hard to know where to start, and that's one of the main problems, is knowing where to start, actually, for something that's so diverse as osteoarthritis is.
If I can turn now maybe to your own research in UCD, your research team there have found a difference in the DNA of people living with rheumatoid arthritis compared with those without it. And, we showed that people with this difference made less of a protein that was then called C5 or 30. What does C5 or 30 do?
Well, it's very interesting, and there have been developments in this, which I'll have to talk to you about sometime soon. What we found was a marker, so a variant, a DNA variant, that was linked with risk of developing rheumatoid arthritis. And then I did a study where we found that variant caused the rheumatoid, was associated with the rheumatoid being much more severe vis-a-vis damage to the joints that we measured on X-ray, hands and feet joints. So the variant was in the gene for C5ORF30, which is a protein that we didn't know what it did, completely unknown. So we've about 20,000 genes and maybe 50,000 proteins. So this is one of them, and it just had that name. No one had done any studies on it. And to cut a long story short, we were able to show that C5ORF30, the catchy name for this, was present in 2 cells in the rheumatoid joint that are associated with inflammation and damage to cartilage. And if we turn C5ORF off, it reduced the potential for a cell type that Ursula does a lot of work on, called the fibroblast, which grows over cartilage in the joint and erodes into it and destroys the cartilage. And if C5ORF is reduced in those cells, they become even more aggressive. So it seemed to us that low levels of C5 was the underlying basis for this association. It may not be, actually. We suspect not. It's a nearby gene. It's something completely different, but we're doing the experiments on that at the moment. So genetics. It's complicated, actually. There have been some criticism of genetics. There have been lots of millions, tens of millions of euros invested in genetics across Europe, hundreds of millions. Hasn't possibly given us great insights into disease pathogenesis. It's just identified a few novel proteins. It's mostly told us what we knew. We knew in rheumatoid that it was the immune system, and that's what genetics have told us. So there are just 1 or 2 hits that have been unusual, and this one we've stumbled on in chromosome 5 is unusual in that it's not the immune cell, it's the fibroblast is in the cells, this type of cell, as I say, that Ursula does a lot of work on, that is different in patients according to their genetic profiles, and they get more severe disease if they have a particular genetic profile based on that variant, that's just small variant in that gene.
We're going to take a short break from our conversation with Gerry and Ursula to take our regular look at popular beliefs about arthritis.
Thanks a million, Gráinne. Today we're going to examine cracking knuckles and the belief that it brings on more pain, more stiffness, and even arthritis itself. As part of our regular look at popular beliefs and understandings about arthritis, we're asking, does cracking your knuckles give you arthritis? Growing up, we were told not to do it. Was that because it was bad for our joints, though? And if it is a myth, where did it come from, and how did it become so ingrained in the public consciousness to be almost accepted as fact? Later, we'll speak to chartered physiotherapist Ciara Mooney, but first, we're off to Deer Park in South Dublin to hear from Niamh Hogan and get the opinions of local people on cracking knuckles.
No, I don't think that it does cause arthritis, but I have heard of that, so I think it's just a myth, but scientifically it doesn't.
I think that's absolute nonsense, old wives' tale.
Yeah, I suppose I get a little bit nervous about it sometimes. I crack my knuckles a good bit, and only recently I've kind of noticed that my hands are a little bit tighter, and I wasn't sure if it was overusing the phone or if it was cracking the knuckles, but it's something that I definitely think about.
Cracked knuckles, is that what you call them? Yeah. Never heard of them.
Always told that you would have arthritis, and you're giving yourself arthritis if you do crack your knuckles. And so we didn't, but as I say, it was in the family in any case, so it was on its way eventually.
Yes, I've heard that, and most of what I heard, my younger sister does it all the time, and my mother used to always say, you'll get arthritis, but we're convinced, no, it doesn't lead to arthritis, so that's what I feel.
Okay, I never heard this about this. I have arthritis myself. I don't know if it's true or not.
Today we are talking to Ciara Mooney of Fearon. Ciara, we heard some interesting thoughts and opinions there during the Vox Pops. So can you put us a bit straight on this? Were our parents protecting our joints from arthritis, or did they just not like the sounds as we were growing up?
Well, Peter, I think they probably came from a genuine place. I think the sound probably puts people off. And I think people are generally worried about it. And I actually get asked this question quite a bit in clinic. I think we have to pare it back to have a look at what evidence do we have with whether or not this causes an increase in the risk of arthritis. And this is actually quite interesting because it goes back a bit of a while. And there's one study published with one participant called Daniel Ungar, I think his name is, and his mother and his mother-in-law kept at him, giving out about him cracking his knuckles because they said, you'll be riddled with arthritis if you don't stop that. So he said, will I? And this is interesting. You can look up this guy's name online. For 50 years, he did a one-man study to see if cracking his knuckles caused arthritis. So every day on his left hand, he cracked his knuckles twice, and on his right hand, he didn't crack them at all. And after 50 years, he did x-rays of both hands, and lo and behold, there was no difference between the two. So that is our great one-man study to say that the initial evidence is that no, we don't think it impacts the development of osteo or rheumatoid arthritis. Now, since then, we've had a few more studies come on board that looked at, there's one particular study that has 300 participants, 74 of them were habitual knuckle crackers, everybody else wasn't, and they looked at what happened to their joints over time. Now, it's interesting because it did not affect the development of arthritis, but people who habitually crack their knuckles did tend to have less grip strength and some joint swelling, but it did not impact the development in arthritis with one group compared to the other. So I think it's safe to say that the sound is not nice, and that sound actually comes from gas movement within the joint because the joint is surrounded in ligaments that seal it, and there's fluid inside, and it's little carbon dioxide bubbles that give that popping sensation. But that is not connected to actual destruction or change to the joint surface. So no is the short answer, and the long answer is even though it doesn't sound good, we're not connecting it to arthritis. But then it brings up the question is, what does predispose us to arthritis, I suppose? And is that overuse playing into it? And this we do know. We do know the contributing factors to arthritis, and overuse is one of them. Okay? And, what are we defining as overuse? Lots of the research in this area tends to be around the bigger joints, and overuse is those manual, laborsome jobs where, you might have been going up and down stairs all day, you were physically on your feet, and that type of overuse is connected to the development of osteoarthritis, very importantly, because rheumatoid is a different ballgame here. We're talking about osteoarthritis. So activities that involve repetitive overuse type activities over time has been linked to arthritis. But we know that it's not by itself. So there's a few factors, and it's how these factors come together that will make us more at risk of having arthritis than not. So age is the biggest one, which we can't do an awful lot about. Joint laxity is one. So if you had somebody who might have had a real repetitive job, maybe they were they were typing all the time, and they have something called joint hypermobility, and they're getting older, then that person is more likely to have arthritis rather than somebody who didn't do the overuse activity, who didn't have joint hypermobility, and who wasn't getting older. The other big factors are genetics. So that's what you can ask your mother about. Not if she's giving out to you about cracking your knuckles. Ask her, does she have arthritis? Because she might be the start of the problem. And obesity is another one, unfortunately. We have fully connected increased BMI with an increased risk of things like hip and knee arthritis. And interestingly, the other one is trauma. So if you were somebody who might have had a bad knee injury or a bad wrist fracture in the past as a teenager, maybe even, then your body has already done a job at trying to repair that cartilage. So then over time, when the age part of it kicks in, you're a bit more predisposed to having arthritis than the other wrist that you might not have broke. So, I've gone down a tangent there, but there's definitely factors that predispose us to having arthritis. It's generally if we have a few of those that we're more likely, but ultimately, on that list is not cracking your knuckles.
Well, that's the most comprehensive answer I could have asked for, Ciara. Really, really appreciate you giving us that. I think we're all off to Google Daniel Unger and see where he is, but not, of course, before we finish out listening to the podcast. And now I'll hand back to Gráinne for discussion on arthritis research with Professor Gerry Wilson and Professor Ursula Fearon.
Ursula, your research focuses on understanding the underlying mechanisms that drive disease pathogenesis. Can you tell us what that involves?
Yeah, so I suppose the two diseases that I work on mainly are rheumatoid arthritis and psoriatic arthritis, which are autoimmune inflammatory arthritis. And a bit like what myself and Gerry have been referring to, that even though over the last 20 years, the treatment strategies for both these sets of patients have revolutionised, and we have maybe 6 or 7 medications that we can utilise, different ones for both orthopathies, but we still don't know when the patient comes in to a clinic what is the right medication to give them. So they could be put on, a TNF and they fail. They could be put on the next option, fail, could be put on the next option, fail, and eventually maybe the fourth option works, but by the time they get to the fourth option, some joint destruction has happened. So a lot of our research is to try and define from the outset what is the right drug to give a patient. So we're doing an awful lot of work actually in the tissue from the patient, and we look at the individual cells, because one of the, I suppose, where we go wrong an awful lot of time with our research is that we maybe use one cell type or one cell line, whereas actually in the joint of a patient, there's 10 different types of cells, but within those there's about 50 different types of cells that all interact together. So if you don't understand it from that level, you're not going to find out how different patients respond to different treatments. So my work is, trying to find out in the site of information in the patient, one, what's happening in individual cells, and then two, when those cells work together, what specific kind of, inflammatory pathways it turns on, and then can we block those? And so what we're trying to do is do that in, different groups of patients, responders versus non-responders, fast progressors versus non-fast progressors, and seeing is there different signatures, different inflammatory signatures that will define patients. And if we can get clues as to, what would define a patient will respond to this medication as opposed to that medication, we might have a better chance of actually treating patients earlier. And we know the earlier we treat patients, the better chance we have of, them going into remission. But the problem is, and we've just actually, a paper came out over the weekend, so over the last 4 years, one of the things we're finding is that a lot of the medications target one molecule. And we've just published, literally over the weekend, we're finding these cells in the joint of patients that they're called polyfunctional, and they produce loads of actually molecules. So they produce 3 or 4 of the molecules that we target, but we, when we treat a patient, we only target one of those molecules. So when you treat a patient, you might knock out one molecule, but the other 3 or 4 will take over and continue the inflammation going. So that's what we need to really work with patients and work on cells from the site of inflammation to understand. So that's one aspect of our work. Secondly, we're now working on metabolism, which you mentioned earlier on. And so we know that when cells become inflammatory and bad, they're a little bit like Bowl's children. They need a lot of energy, so they go into a sweet shop, they get lots of energy, and then they become bad, as opposed to maybe a child eating, as I would describe it, as a whole grain, pita bread, where it's slow releasing energy and they're good. So that's a little bit how cells work in the joints of patients. So they get their energy really, really quickly, and that maintains the inflammation in the joint. So we found that in patients with inflammatory arthritis, that the metabolism, the way they utilize energy and glucose, differs in the cells in their joint. So what we're trying to do is that if we can target the way they utilize energy, can we turn off the inflammation? And we've actually found that actually when we do isolate cells from patients and turn off the way they utilize glucose, we actually turn off the inflammation. So that's a potential new therapeutic, approach which hasn't been utilized yet in patients with inflammatory arthritis. And so that's the second approach we do. And the very last one, which actually is pre-disease, we've identified, we work on an awful lot of patients, individuals that don't actually have inflammatory arthritis, but they have been identified by their GPs as potentially at risk of getting rheumatoid arthritis. And we've actually found in those individuals that their cells are already metabolically active and actually that is associated with inflammation. So we're actually monitoring those individuals to see, can we predict who will develop rheumatoid arthritis and who won't, and actually can we treat them at the very onset, where again, the earlier you treat, the more chance they have of going into remission. So they're the three aspects of the work we're doing.
And just on that last area of work that you're involved in, how can you identify at-risk patients if they haven't actually got any of the markers or maybe they do?
Yeah, well, they do actually. So what happens is, and so we don't know what starts rheumatoid arthritis, but we know that there's possibly a genetic background, and that with an environmental hit. And the environmental hit, again, we don't know what it is. It could be, there's a lot of association with smoking. It could be a pathogen in your gut, your mouth, your gut, your mouth. I'm trying to think of a third one, but there is a third one. Lungs. So that's the second one. So we get a first hit with this genetic background, and that causes just a change in our cells in our body. And then our immune system picks that up as something foreign, like a virus or a bacteria. So we start to attack ourselves. But we think there's a second hit is needed for those cells in our circulation of our blood to travel to the joints. So in those individuals, what's happened to them is that they have the genetic background probably, they've had that first hit, an environmental hit, but then their inflammation calms down and they haven't got that second hit. But what's left in their blood are antibodies that they produce to try and get rid of the antigen. So actually that's how they're picked up. So they'll go to their GP with aches and pains. The GP can't really find anything clinically, but they'll do a blood test, and the blood test will show that they have these autoantibodies in their blood, and then they're referred to a rheumatologist like Gerry or Doug, and that's when they're identified as an at-risk and somebody to watch. I don't know whether you have anything to add to that.
No, I mean, it is. It's complicated, and the main thing from this is that everyone is different in what starts it. People have spent a long time trying to find the thing that provokes rheumatoid, but it's likely to be different things in different patients. It is, as Ursula says, it's genetics, but it's this thing that we've always thought, this mystery box.
This mystery trigger.
This mystery thing, but it's the environment and how the environment affects how things work, your cells work, your DNA and genes work. And we do know that it does have profound effects on how your genes work, where you live, what you do, if you get exercise, even your error code or postcode. Is a big correlation with how your genes work, amazingly, and so and that's why it's so complicated to work it out, because everyone is different, and that's what we've really realised. It's one of the things genetics has shown us, that to get rheumatoid, two people can have nothing similar, but still get the condition.
So it's that question of, is it genetic? It's
nature and nurture.
There definitely isn't a clear answer.
No. It's what we call multifactorial. So it's lots of different things, but they're different in different people.
Which makes it even more complicated.
That's exactly
and that's actually going back even to what I was saying about that paper the weekend, that the cells are multifactorial. So even one cell is producing loads of proteins. And, we can't give a patient 10 targets to target those, maybe 10 proteins. So it's trying to figure out, can we target something downstream that all those 10 proteins maybe feed into? And that's where metabolism comes into, because all those type of proteins alter metabolic signaling. So if you could stop the metabolism, you could maybe stop the inflammation. It's a bit like, I use this analogy in some of my lectures, where you've got a car and an engine, working car, and you've got a driver. And if you take away the driver, so if it was your car, Gráinne, and I said, no, you're not allowed to drive it, so Gerry could go off and drive your car, and if I told Gerry he couldn't, sure, I could get into the car and drive it. But if I took out the engine of the car, it doesn't matter how many people would turn up, we couldn't drive the car because the engine is gone. And that's where the metabolism fits as well, that it's the engine it is the engine of our cells. So if you can target that in the right way, you might be able to turn off the inflammation.
That's a really good analogy. I think that'll help people understand. Yeah, absolutely. I mean, the area you're involved in is translation and research, and you touched on that earlier, but what exactly does that mean?
Yeah, so I've got a bee in my bonnet about this phrase, and it's a bit translational research, bench to bedside, and I suspect Gerry has the same bee in his bonnet about it. So an awful lot of people throw that word around, and so it's, I could actually go to Gerry and say, can I get, blood from 20 patients, and I could go and in the lab and look for a protein, a marker of, that I think is inflammation, a marker of inflammation. But an awful lot of researchers stop there and they publish a paper saying, oh yeah, this protein is higher in this group of 20 patients compared to healthy controls. But within that 20 patients, as we've alluded to throughout this whole podcast, they're all different. All the patients are different. So it's a circle. You have the clinician who, assesses the patient, discusses the disease with the patient, discusses the treatment. Then the patient, consents if they wanted to get involved in a research study. Then in the lab, we examine either markers in their blood or in their tissue. But the only real way to do translational research is that when you measure the level of a marker in a patient's blood or tissue, you relate it back to the inflammatory status of the patient, because that's the only way you're going to figure out what's different between patients and how you can treat patients. So it's a full loop. You have to interpret your data in relation to the patient you got the samples from. Really important.
So that's right. So it's conventionally been said to be bench to bedside, but it's also important to realise that it's bedside to bench.
Yeah, sure.
And so its clinical observation can lead to research. And there are lots of examples of that. The finding of one of the treatments is rituximab that we use for a rheumatoid. Now that had been used for lymphoma for many years. Yeah. And it was just that it was noted there was a patient in London treated with rituximab who had lymphoma but also had rheumatoid. And it was observed clinically that the rheumatoid responded very rapidly to rituximab. Because what rituximab does was predicted to be unimportant in rheumatoid prior to that. Surprisingly, it was, and this came out of just a clinical observation. Yeah. So it was really, as you said, it's a loop. But you can start in the clinical bit and just observe something, and then take it right round back into the to the bench and do the experiments and work out why that observation occurred, and then take it back.
But it's even if you go back to TNF stands for tumour necrosis factor. It wasn't it was more in the cancer field initially, and then the Kennedy Institute took tissue from patients, and it wasn't even one of their top targets. And then when they put it into the tissue of patients, that's when they saw that actually this protein is actually the hierarchy of a lot of the inflammatory proteins in the joint. So we wouldn't have TNF, which really started the revolution in treatment for patients, unless we had patients, consenting to be involved in research.
Yeah, that really nails it, doesn't it? Yeah. I know another area that you've both worked on in recent years is in relation to Down syndrome associated arthritis. And this is obviously something that Arthritis Ireland has supported for a number of years as well. Can you tell us about that research and the findings?
Yeah, so I'll start off maybe just about the clinical picture, and then maybe Ursula about the research. So really the research that you funded was initiated with Orla Colleen in the Crumlin Hospital, and it was undertaken as a PhD by Charlene Foley. And what Charlene did was she actually went in her car and went out across Ireland and visited families and examined children with Down syndrome and examined for inflammation in the joints. And as far as I remember, it was around 500 children that Charlene visited and examined, and she found at that one time for each child evidence of inflammatory joint disease in 33 children. So I think that's about 6.6%, as opposed to the risk of juvenile idiopathic arthritis is about 0.1% of children. So there is this vast excess of inflammatory arthritis that was not recognised in children with Down syndrome, and I think it was just missed because there were other issues, perhaps other health-related issues, and it wasn't found. And we do know that it's more difficult to treat than juvenile idiopathic arthritis. For example, the standard, go-to drug in inflammatory arthritis in children and in adults is methotrexate. But in Down's arthritis, it causes kids with Down's arthritis. It causes a lot of nausea, and that inhibits its use, and so they have to be, cycled on earlier to other treatments. So there was some very interesting. It was a lot of hard work. It was a good project, a lot of hard work, but it just shows you the benefits of doing. This was clinical observation and just spending time in the car, going around and examining 500 plus children with Down syndrome, and finding 33 at that stage with Down's arthritis. As I say, and that's 6.6%, which is very high, right? So, Ursula, do you want to tell us about the research?
So I came in after Charlene had and Orla had identified this vulnerable group of children, and so. Actually, if we go back to the beginning, when Orla first noticed it, she didn't know whether it was just by chance that every time she saw a child with Down syndrome and inflammatory arthritis, that the inflammation just always seemed to be bad. There wasn't this spread of mild versus moderate versus severe. It just always seemed to be bad. So she didn't know whether it was just that children with bad arthritis was being referred, or whether this was a different type of disease. But actually then when they went out and went around Ireland, they actually did find that actually the incidence of severe inflammation in these children was higher than the general population. So then that's where I came in, where we wanted to understand, well, is there something different in the joints of these children that's actually driving this more severe disease? So we did, and I suppose I talk an awful lot about, patients getting involved in research, but actually this is the one that I'm really passionate about, because it was young children and their parents that consented for us, to have samples, of some blood and some tissue. And actually it really has shown us huge difference. And I think you've seen the pie charts that I show.
Yes.
And I show a pie chart where nobody on the podcast can visualize it, but basically we looked at immune cells in healthy controls, children with juvenile idiopathic arthritis, children with Down syndrome, and then children with Down syndrome and associated inflammatory arthritis. And the healthy controls have about 3 or 4 colours. Then we get to JIA, which has about 6 or 7 colours. So the more colours, the badder the immune cells are. Then we get to Down syndrome, and they're a little bit worse than the healthy controls, and we know they have immune cell dysfunction. And then you get to the children with Down syndrome and inflammatory arthritis, and they've got about 20 different colours showing that the immune cells are very bad. And then we also looked at the synovial fibroblasts, which Gerry referred to earlier on, and these are the cells that actually are in your joint, and they invade into your cartilage and basically release proteins that eat away at your cartilage. And we found that when we grew them in the lab compared to healthy fibroblasts, they were much more invasive. Again, they were really metabolically active. They were switching their metabolism. They wanted lots of glucose to keep them active. And every measure that we did in these fibroblasts of inflammation, of invasion, which allows them to invade into cartilage, was much higher than your healthy controls. So these children have those immune cells that, like I was referring to earlier on, that are producing lots of pro-inflammatory mediators, but they also have these fibroblasts that invade into the cartilage at a much more aggressive rate than normal fibroblasts, even in children with JIA. So actually, it's a very severe form of inflammatory arthritis, and as Gerry said, they don't tolerate methotrexate. I know already that the next line of treatment is TNF, and Orla would say that probably that's not great either. And she would like, I think, or the consensus, possibly we need to be looking at other therapies for these children that we have identified already, that are being used in the adult population as well, to see what works best. I mean, they are, in juvenile arthritis, there's about 7 different subtypes, where there's really there's about 3 subtypes, but really one main subtype in DA, and I think it's kind of along the lines of psoriatic arthritis.
So it's a kind of a subtype in itself.
It is,
yeah. So actually, but I think that's really amazing, that story, because it went from a clinical observation, by Orla, where they literally went on the road in their car, And, you know, now we know both from a clinical point of view and a scientific point of view that actually this is a new disease. And there's now an awful lot of focus on it. And those children, there's more awareness, and those children are now being referred to Crumlin Hospital. So already there is an impact, you know, a clinical impact, you know, because an awful lot of time from a scientific point of view, it could take 10 years, 15 years to actually see an impact. Whereas that within 3 or 4 years had an impact that children were actually being picked up much earlier, and Orla was actually seeing them much earlier. So that's an amazing story of how maybe Arthritis Ireland funded some research that actually has already had an impact in the clinic.
Yeah. So children are being picked up quicker. Children with Down syndrome are being picked up quicker. So the remaining kind of piece of the puzzle is looking at the therapies that are going to really target this, to bring it under control.
And using them early. It's really about, as much as any of these inflammatory conditions, it's recognising as early as possible. It's easier to switch things off and treat things early. And, you know, things do become more complicated the later the condition has, the more it's evolved. And so it's the holy grail of treating inflammatory arthritis is to get there as early as possible and try and switch off inflammation as early as possible, and then to control it at that stage, because inflammation equals damage in the long term. It's a very simple formula that I teach the medical school students. The amount of inflammation over time, that equals joint damage. And if you switch off inflammation quickly, joint damage doesn't really happen. So that's the key thing. That's the key thing now. A simple observation in Down's arthritis, that it's so prevalent, means that kids with Down's arthritis have to really be examined. Their joints has to be, and that's a worldwide thing. Hopefully that will be pursued. Yeah, and it's actually now, so Orla's now also working with, and I'm working with Professor Eleanor Malloy, who's, you know, the professor of pediatrics in Trinity now. So we're also working with her now on that story. And I've got a PhD and a postdoc working on it in the lab now, and we've actually identified a new signature in these children, which I'm not going to say what it is yet, just we need more confirmation, but it's looking like this signature could be targeted as well. So so watch the space. Yeah, I might turn to our last subject now, which is the Arthritis Research Coalition, just to talk a little bit about that. So this was established a few years back. The objective being to coordinate clinics and hospitals across the country to develop, a biological registry and biobank of patients in Ireland with rheumatic diseases. Why is it important? And tell us a little bit how it's going so far. Well, as we've sort of we have discussed, translational research and moving from the bench to the clinic and back involves having access to well characterised groups of patients with different conditions, having detailed histories of who they are, what they do, their previous medical history, their treatments, and their condition. How active it is at any one point, and linking that up with samples, and it's mainly blood samples that we can get DNA. We can have a sample where we can look at how active the genes are, and also serum from the blood, so we can measure proteins in the serum. And so if we have that as a repository, it's then it's much easier to be active and continually active and think up relevant experiments that we can undertake to try and understand better the causes of different types of arthritis, inflammatory rheumatic conditions, possibly also look at response to treatments, and markers that will help us predict who will get severe types of the different conditions. And those are the key things. So having biorepositories allows us to really take our research to the next level, because our research, which is translational, does rely on access to samples, and it takes a long time to get samples because you have to go through a whole process of, for example, ethics. Which takes a long time to get samples, but we've got the ethics for recruitment for all the common rheumatic conditions across many of the major hospitals in Ireland. And so that's really the key. It's that we're primed to do research, and we have this Arthritis Ireland funded repositories of different conditions, including Down's arthritis, juvenile arthritis. Families with arthritis are different types of inflammatory rheumatic conditions and types of arthritis, as well as rheumatoid. We're doing gout, systemic lupus and psoriatic. And so we'll have all of them. And hopefully we can continue that. But it does allow people to do research. And it's a good selling point for people who want to come back to Ireland and do research, for example, or move to Ireland. To say that there is this repository that is being developed nationally that people will have access to undertake research, relevant research, funded research. So that's one of the reasons. It's really important to have good infrastructure for research. And this is it. It's not about any one individual. It's about, the community. And that's really why it's important to develop this. As we've discussed, good research in rheumatic diseases, and the major advances in rheumatic diseases have been reliant on patients helping and contributing to studies, whether it's just histories, but also, the examination, and also giving samples that we can use to spot differences or ways of predicting things, response to treatment or severity of the disease. The more data we collect, the better, the more useful the samples are. And part of ARC was developing this sort of bespoke online data and capture system that can be used anywhere, and it's all ethically approved. It's easy to get the information out the back end, which is a key thing in doing research. So that's what it is. It's a sort of thing for everyone. But it's owned by no one. It's owned by Arthritis Ireland and the community, really. And hopefully it will attract and support young investigators to come along and undertake research, because they will have access to samples that can be used for state-of-the-art research studies.
And of course, the central point of it as well is having patient involvement.
Yes.
Because without that, it doesn't happen. So for patients listening, that might be something again that they might consider getting involved in it.
And hopefully we're developing the website.
It's on our website, arthritisireland.ie. There's further information about ARC, so patients can look that up as well, about how they might get involved.
Yes.
Because it's really important about building up large numbers. Because if you've got small numbers where you're trying to decipher what therapy works for what group of patients, if you've got small numbers, and that's probably the problem so far, you've got small numbers, you won't get a definitive answer. And that's possibly because all different researchers across Ireland were working. So you could have maybe 20 different groups working on rheumatoid across Ireland, but we were all just working on our own little group. Whereas if you combine all those groups, you have a much bigger power to identify something that's different between different groups of patients. So I think it's having the whole of Ireland working together for starters. It's large numbers, it's patient involvement, and as Gerry said, it's everyone can work in it. So once we build up this large data set, if somebody has a research idea, they can put forward a proposal to access the data that's there or access the samples that are there if they have got a good proposal. So that's why it benefits everybody. Actually, everybody. The clinicians, the patients, the scientists, everybody.
And last question for you both. What does the future hold in terms of rheumatology research? And, what do you think that future might look like for patients on the foot of that research?
Well, I'm kind of weird, and this is another one that's thrown around the place, personalised medicine. So I suppose what you would love for patients is that they arrived into their GP or their rheumatologist, like Gerry here, and literally you could do a quick screen, little blood prick or something, do a quick screen, and it will tell you, one, what form of arthritis they have, two, based on the screen, what medication this patient will go on to. So we're a fair bit away from that yet, but at the same time, there's these new, really amazing new technologies that are now out there that we don't have them in Ireland, but we're utilising them with industry partners at the moment. And basically we can look at all the genes in single cells and then look at two cells beside each other and see how they're interacting. So that's going to give us huge information on an individual level as to what's going on in individual patients. But at the same time, we can't do biopsies on everybody. It's just not practical. That will give us the answers possibly, but in a routine clinic you cannot do. So what we want to do is find out what's happening at a single cell level in the joint, then translate that to try and find a surrogate marker in the blood, and for that to be used as a marker that we can use actually in the clinics. So that's the ideal where we want to go. And in some aspects, there's some aspects where we are utilising personalised medicine. So we know that, if you've got high ACPA, if you've got, a certain pattern of cells in your tissue that produce antibodies, the likelihood is possibly you should go on rituximab, what Gerry was talking about earlier on. But we're still not there really at predicting who goes on to what medication. But that ultimately is the future.
Yeah, I mean, it is actually, it's a big area of research. And other things in the future will be to use, and I'll just throw a few things out. We do have to get better. We do have to improve our research and develop research into some of the needs areas. Osteoarthritis, it's just huge, and we've just nothing that touches it. Gout is another. Crystal arthritis is so prevalent. And we really need to know quite a lot more about the causes, underlying causes, and the best treatments for gout and other types of crystal arthritis. And one thing another area that's sort of interesting to me in some of my research is the use of the new technologies. And so when I give talks for Arthritis Ireland, I'm frequently asked about families.
Yes.
And so there is the new sequencing technology and the ways of looking at analysing the data which have developed. But it is actually working, and it's selecting families, that where you see a condition appearing, whether it's in one generation or it's coming down the generations. And that's really from from bedside. So it's clinicians recognizing that there may be a genetic cause of a condition, and then we can try to use this new genetic profiling to map, to find out what the mutation is. And if we know the mutation, we can really get personalised. It's a small group of, it's not like the overall rheumatoid population, but there are lots of families out there with these conditions. And the hope of personalised medicine is really, it's much easier in those conditions because we know it'll be a single gene issue, and targeting that will be a way of treating those families, and that's what we're trying to do at the moment. We have been collecting families and using these new technologies, and we're doing it in a childhood condition, inflammatory bone condition, and we're finding a subgroup with particular very rare. Well, it's about 15% of them have mutations in a particular key gene in the immune system, interestingly, and so we've just sort of been getting that data recently. And so using the new technologies in families will be, I think, very useful, and it will actually be particularly useful in personalised medicine, because it's one single thing that there'll be a defect in or a problem. It'll be overactive or underactive, and we can use a drug to really correct that, hopefully. So that's a particular area, of interest of mine anyway at the moment.
Well, on that positive note about the future, I will say thank you to you both. Thanks for your time. Thank you, Gerry Wilson. Thank you, Ursula Fearon.
Okay,
Thank you, Gráinne.
That's all from this episode of Inflammation Nation. Don't forget to subscribe on Apple Podcasts, Spotify, or wherever you get your podcasts. For further information about arthritis, you can visit our website, arthritisireland.ie, or contact our helpline on 0818 252 846. See you next time. Inflammation Nation is supported by Pfizer.